Hello and welcome to Facebook Live and YouTube streamed versions of SoapBox Science Nottingham. SoapBox Science is a series of public engagement events funded by the Zoological Society for London and supported by the STFC. The premise of the events is that women stand on soapboxes, actual real soapboxes, and talk about their science. Usually these events would be run in person, and that's what I'm talking about with the soapboxes. And I have fond memories of having done the event several times myself, standing in a rainy market square or a busy shopping centre and talking about my science. But due to the obvious, we've gone virtual for this event. So we're going to try and keep all the magic and all the excitement that we usually would have, but bringing it to your living room, your mobile phone screen, your computer screen, and bringing you along with us for the excitement. This is also the first time that SoapBox Science has come to Nottingham, and as such, it's our first virtual SoapBox Science Nottingham, so we're really excited. It's also a collaboration between scientists at the University of Nottingham and Nottingham Trent University, and I'm introducing my colleagues here, Helena and Zara. Helena, would you like to introduce yourself?
Hi, good morning, everyone. I'm an Anne McLaren Research Fellow at the Food Water Waste Research Group in the Faculty of Engineering here in Nottingham. And this is my third time organizing SoapBox Science. I never had the courage to stand in the soapbox, but I was member of the organizing team in Hull back in 2016 and 2017, and it's so good to be part of this group and organizing here this year in Nottingham. So I had the pleasure to know more about the research of so many amazing women, and I think today will be an excellent show of what the science women are doing here in East Midlands.
Thank you. And Zara?
Good morning, everyone. I'm Zahra Al-Ahmadi. I'm a senior lecturer in advanced therapeutics at the School of Science and Technology at Nottingham Trent University. It's really a pleasure to be part of organizing this SoapBox Science Nottingham. I had the privilege to be presenting to present before as in SoapBox Science Manchester event in 2016, and it was really an exciting moment for me that I still really remember talking to the public about my science, and also here to the other women scientists presenting their work. And I'm really looking forward to the event today, to hear from the lovely women scientists in Nottingham, and also share with you the exciting work that they do.
Brilliant, thank you, Zara. And I'll introduce myself as well. I'm Rebecca Dewey. I'm a postdoctoral research fellow in neuroimaging at the University of Nottingham. So I've got a background in MRI physics, and now I mostly look at the brain and how the brain handles hearing. And as I said, I've presented at SoapBox Science a few times before, at Grantham, Lincoln, and at the Blue Dot Festival. But what we're here to do today is to listen to 10 fabulous speakers that we've got lined up today. So Helena is going to introduce our first speaker.
Yes, so we are starting this morning with Dr. Amanda Butler from the University of Nottingham, and her talk is about lungs. So when lungs go wrong.
Hi, good morning. Pleasure to be here at this virtual SoapBox Science event today. My name is Amanda Butler, and I'm going to talk you through some of the research that I do into lung structural changes in respiratory diseases. I'm going to talk to you today about two respiratory diseases, one that you've probably heard of, asthma, and another that you might not have heard of called lung fibrosis. And I'm going to finish my talk today talking to you about some research that I've been doing this year into coronavirus. Coronavirus with me. So to help me do this, I've bought a balloon which is going to help me model the lungs. And our lungs are really remarkable organs. They're the site of gas transfer into our bloodstreams. This is where oxygen that we breathe in is transferred into our bloodstream, and conversely, CO2, carbon dioxide that's in our bloodstream, gets back out into the air. They are large sacs of air, also known as alveolar spaces, and the air travels into this air sac via airways. This starts in our trachea and it travels down and branches out into the air spaces of the lung themselves. I'm going to start talking to you about my asthma research, and asthma is a disease primarily of the airways, and that is these tubular structures that feed air into the air spaces. Now, in a normal lung, air passes quite freely through these airways to fill the air spaces, like this. But in asthma, what happens is that the airways start to narrow and to swell, and the muscle around the airways contracts, and it makes the lumen, the area of the airway, much smaller. So air struggles to pass through the airways into the air spaces. I'm going to mimic that with an elastic band. So I have my lung, my elastic band. I'm going to tie the elastic band around the airway, so the opening to the air spaces. And then you'll see that it's actually suddenly very difficult for me to get air into the balloon. It's really, really quite hard to blow air into that lung, and you might have heard noises. And that's really mimicking these episodes of airway narrowing that are characteristic of an asthmatic, an asthma attack. But the lungs of asthmatics also change in structure, and some of these structural changes involve the airways. So the muscle that surrounds the airways that is actually driving that contraction, that airway narrowing, in asthma gets much thicker and stronger and can contract with more force. The airways of asthmatics also often have scar tissue around them, which makes the airway much stiffer, and that also can impede air travelling through the airway. Another change that some asthmatics have is that their mucus cells, the cells that produce mucus within the airways, start producing excessive amounts of mucus, and that can start blocking the airways and further obstructing airflow. Now, the second lung disease that I want to talk to you about today that also has lung structural changes is called lung fibrosis. And unlike asthma, where the disease is primarily found within the airways, lung fibrosis is a disease of the air sacs themselves. And this disease is characterized by the development of scar tissue around the air spaces themselves, around these alveolar sacs. And to mimic that, I have a fruit net. This used to hold some lemons. I'm going to put my lung inside, mimicking this scar tissue that builds up around the air spaces. And I'm going to try breathing again. You can see I can get air into the lung, but the balloon is very restricted in its size. The scar tissue is so stiff and so dense that it restricts how much air you can get into the lungs. And that has a very severe impact on people with this disease. They struggle to breathe, and it's progressive. So this scar tissue just keeps developing and getting thicker and more widespread throughout the lungs until eventually lung fibrosis is a fatal disease. So people succumb to respiratory failure. Now, these are two very different diseases. One affects the airways, there are many different differences between these diseases, but they both involve structural changes to various parts of the lungs. What causes these changes? Well, my research over the last 15 years and many other people's research around the globe has highlighted a potential role for a protein called TGF-beta. Now, TGF-beta can increase muscle thickness around airways. It can increase scar tissue formation, both in the air spaces themselves, but also in the airways. And it can also have quite dramatic impact on cells that produce mucus in the airways, and it can increase mucus production. So you can see that this is really quite an important target when we're thinking about candidate proteins that might impact structural changes in the lungs. Now, what's really interesting about TGF-beta is that it's produced and released from pretty much every cell in your body, but they release it in an inactive form, and they surround themselves in huge amounts of inactive TGF-beta. And it's only through understanding how that inactive TGF-beta becomes activated that we might be able to understand how structural changes such as scar tissue forming or increased muscle around airways might occur. Now, in asthma, I've been able to show that when the smooth muscle around airways contracts, causing that airway narrowing during an asthma attack, the muscle cells themselves can activate TGF-beta. And that creates a really direct link between something that's happening during the asthma attack, i.e. the airway getting narrower, and the development of structural changes. So the airway gets narrower due to muscle contraction, and that causes activation of TGF-beta, which then causes structural changes in the lungs that has a negative impact on asthma. In fibrosis, it is well known now over the past decade or two that increased stiffness of tissues can impact TGF-beta activation. So the more stiff a tissue, the more TGF-beta gets activated. And in lung fibrosis, this scar tissue is stiffening the lungs. So you could see how this is a link between, this might help explain the progressive nature of lung fibrosis. So you get some scar tissue and that stiffens the lungs. That causes increased TGF-beta activation, which causes more stiffening of the lungs, which leads to more TGF-beta being activated and more. So you get this perpetual cycle, which might help explain why lung fibrosis is as progressive and irreversible as it is. So finally, I've got 2 minutes left just to move on to the work I've been doing this year. So this year has been a very different year for me, as it has for pretty much everyone probably around the world. When the pandemic really hit the UK in March, I joined a group of colleagues who wanted to understand how SARS-CoV-2, that's the virus that causes COVID-19, might affect the lungs. We've been particularly interested in the SARS-CoV-2 spike protein. So coronaviruses have these characteristic spikes, and we've been interested in how the spike protein of this virus might impact the lungs. We've been able to show that these spike proteins can bind to 3 different receptors in the lungs, and these receptors are very widespread within the lungs. And interestingly, 2 of these receptors are capable of activating TGF-beta. What we've also found when we've looked at lung tissue from patients that have sadly died of COVID-19 is that they have evidence of scar tissue around their air spaces. So they've got scar tissue very similar to that we see in lung fibrosis. So somehow SARS-CoV-2 virus is causing scar tissue to form. So we've looked at whether the spike protein itself, when it binds to these receptors, can cause TGF-beta activation. And actually we found that it doesn't in our experimental systems. But there are still lots and lots of ways in which infection with SARS-CoV-2 could lead to scarring of the lungs. And we and many groups around the world are really trying hard now to try to work out what those reasons and those mechanisms might be. So thank you very much for listening. It has been a pleasure to talk to you today at this event. I will happily answer any questions that you have. Thank you again.
So we'll now answer some questions to Amanda. Let's see if we can get, yes, we can get Amanda here live.
Hi, good, thank you.
Hi, Amanda, how are you today? I'm good, thank you. Thank you so much for participating in this year's SoapBox Science event in here at Nottingham. I just wanted to ask you a little bit about your career. So can you just tell us who inspired you to get a career in science?
So I always loved science when I was at school, and my A-level science teacher sort of inspired me. I always had a real interest into human diseases. So when I was looking at degree programs to follow, I was really interested in one called biomedical science, because that covered a lot of human diseases and the mechanisms of human disease, which I studied for at Nottingham Trent University. And then it was my final year research project supervisor who really encouraged me to go and do a PhD and follow an academic research career. So then I applied for some programs and loved a program at the University of Nottingham in respiratory diseases.
Okay. Also, now as a question about COVID-19, because it's really what people are worried about. So you study asthma and also you're studying COVID-19. So are you also studying how both diseases can affect each other?
Not currently, but I'm sure that's what will come next. So originally in March, when lockdown happened, all of our non-essential research was stopped. So we had expertise in respiratory disease, so it made sense that we started looking at COVID-19. So initially we were just looking at the effects of the viral infection themselves, but yeah, there's still much to know about COVID-19.
Okay. Yeah, I think we have a bit of delay between the live transmission and the comments. So I really don't have many questions from the audience about your talk, but maybe we can talk a little bit about also what's the most fascinating aspect of your research. What do you really think it's the aspect that makes you wake up every morning eager to go to work?
So I as I say, I've always been interested in human disease, and what I really like about the sort of research that I do and that we do in Nottingham. Sorry, you can hear my 1-year-old in the background. I love the fact that what we do on the bench in the lab eventually translates into patient benefits. So in Nottingham, in the Division of Respiratory Medicine, we work really, really closely with clinicians in the hospital that are treating patients with these diseases. And it's fascinating to watch early discovery science then be translated into patients and into potential new treatments and clinical trials and things like that.
Okay, yeah, it's true. Okay, so if you would do a recommendation to a female PhD student considering a career in academia, what would you recommend her?
Oh, and follow it. I mean, if you're interested in research and learning things that nobody else knows, I that's another thing that I love about my job, is that I'm constantly finding things that nobody else has found yet and trying things that nobody has tried to do. So if you're passionate about science and you're interested in science, then definitely follow a career in research. It's great. I've traveled, I've been and worked abroad in my career. And I think that there's sometimes a criticism that maybe academia doesn't mix well with family life or that work-life balance, but I actually find it very flexible, and I think it works. It certainly works for me. Even if that's live streaming with a 1-year-old.
Yeah, I think so I'll just try to reach to our audience. Please ask your science questions in the chat, both in YouTube and Facebook. We are reading your comments, so if you have any questions, please let us know. We are happy to ask them live, and you can get some insight from women scientists on live. So please just let us know what you want to know about any of the themes that we are talking this morning. Yeah. So Amanda, wow. I don't know what.
Well, I was just going to say as well, if anybody has any questions after the event or later when it's, not my turn, you can @ Amanda Tatler on Twitter to me, and I'll happily answer any questions anybody's got. If you suddenly think later, oh, I wish I'd asked that, then just give me a shout on Twitter.
Yeah, that's great to know. I think, yeah, because the videos will be available. So if people reach these later on, they can also get your contact and then contact you directly. Yeah, that would be great. Okay, so I guess we are moving forward to our next presenter, and this is Andrea Konta. She is a PhD student at the University of Nottingham, and she will talk to us about 3D printing. And the title of her talk is "How Can Invisible to the Eye 3D Printing Help You?" So let's see what Andrea has to tell us.
Hi there. I'm a PhD student at the University of Nottingham, and my research focuses on 3D printing. So I'm sure everyone has heard of at some point of 3D printing, either via news, online, or some other type of media outlet. But what is exactly 3D printing? So it's the process of adding layers of materials on top of each other to manufacture a structure. And that's why many times 3D printing is referred to as additive manufacturing, especially in the research and industry world. So there's many different types of printers that use different techniques and allow for different materials to be printed and to be manufactured. And quite a few people are familiar with extrusion-based printers, where a plastic material like PLA or ABS polymer is extruded through a nozzle to manufacture your part. So an example of this would be these two chess pieces here. So these are produced with this type of printer, and they're made out of PLA polymer. So you can see them here. You have different colors and play around with the designs. So these type of printers, there's quite a few that are, at affordable price, and people can buy them, have them at home, and have a bit of play around with them. But there's obviously many other different techniques, not based in extrusion. You can use laser, UV light, other type of things to manufacture different materials. So you can also print metals. And here would be an example of 3D printed lattice structures with metal. There you go. You can also have other types of designs, like little chess piece as well. This is also printed in metal. But you can print many other materials. You can print ceramics. You can print other types of polymers. It's not these two that I mentioned. And do like something like this Lego inspired piece. Or you can even print cells in what is called bioprinting. So 3D printing is allowed to print designs that wouldn't be possible to produce with maybe the current manufacturing techniques. And there's many different industries that have started to use it and show how versatile it is. So for example, the aerospace industry, electronics, medicine and pharmaceutics, the food industry, or even fashion. And additive manufacturing allows to produce parts at different resolution and sizes. So my research, for example, focuses on printing at the micro and the nanoscale. So very, very tiny parts that you can't really see with the naked eye. So you need a microscope to see them. And this is done by a technique called direct laser writing or two-photon polymerization, where what we do is that we have a liquid resin, usually made of a monomer or polymer that reacts to light. And we solidify that liquid resin into the design that we want by exposing it to laser light. So an example of something similar, but that you can actually see, it would be this Lego inspired piece that I showed before. So this is also produced from a liquid resin that you solidify, exposing it to light. But obviously these type of printing produces parts that are way bigger than what I work with, which is like very, very tiny ones. So micro 3D printing can be used for quite a few different things. For example, for metamaterials or even cell culture. So metamaterials are materials that are engineered to exhibit some specific properties that the natural material does not have. So for example, with micro 3D printing, while you print, you can design a specific structure inside the material that would allow it to manipulate the way that light passes through it, and that can help us to make it seem invisible, for example. That's a good property that you can do with 3D printing that the material doesn't have naturally by itself. Another thing that we can do is related to cells. So when the stem cells grow and they turn into specific heart, lung, or bone cells, there's many different things that guide them to become that specific cell type. And one of these things is their environment and the topology of the surfaces around them. So what we can do with micro 3D printing is create this specific environment and topology, and that would allow us to determine what specific cell type we want to obtain. So as you can see, there's quite a few nice things that you can do with micro 3D printing, but obviously it also has its disadvantages. And one of them is that we don't have yet a very broad range of library of materials that we can use with this technique. Therefore, there's a need to develop more materials for this printing type. And that's what my research focuses on. To be more specific, I focus on developing new biomaterials for this printing technique. And biomaterials are natural or synthetic materials that can be used inside the human body without causing it any harm. And they're usually used in medical applications to, like support or enhance, for example, some damaged tissue or a biological function, or even deliver drug inside the body. So therefore, my work is to produce new biomaterials that are reactive to light, so they can be used with direct laser writing. And once we have, developed and produced those materials, and we make sure that you can print them well, they manufacture structures properly and all that kind of stuff. And, I say it fast, but it takes some time to get to that point. Once we reach that point, we run specific tests and cell toxicity studies on them to make sure that they are not toxic for our bodies. Once we have made sure of that, what I want to do with the new biomaterials I produce is to print a small implant that can deliver drugs over several months inside the eye. And what I want to do with it is to help patients improve their symptoms with eyesight problems. And that's it from me. Thank you very much for listening.
Okay, so I would like to remind our audience to please post your questions in the chat, because we'll have opportunity to talk live with Andrea. So please put your questions both on YouTube or Facebook, and we can read them and we can ask them live to the scientists who are participating today on SoapBox Science. Hi Alice, how are you today? Good morning. Thank you so much for participating in this SoapBox Science event.
My pleasure.
So we really liked your video. And I would like you to talk a little bit more about 3D printing. What do you think is the most exciting aspect of 3D printing? What do you think it's really
Well, there's many things, because as I said, there's many different types of 3D printing. So each area has its own exciting thing going on, basically. But specifically for my case, for example, because I focus more on like medical and pharmaceutical applications, right? I think it's also the shift to being to get to a type of medicine that is more personalized. I think that's very exciting, because you can get very different types of treatments, implants, drug delivery systems, cell type of patches, everything that can be very personalized to each patient. So then you can really focus to treat each person to their own needs, instead of just doing batch type of medicine, which it's okay, but it's definitely not as good as going individualized to what each person needs. So I think that's quite exciting.
Okay, yeah, it's true, because then you can target different diseases and customize treatments for different people. Yeah.
Yes, and it's more like each disease is even of the same type of disease, it will behave differently in each patient, right? So then you can tune it to that.
Okay. So what is your favorite equipment for 3D printing? What do you like to use in the lab?
Well, I mainly work with my type of straight printer, which is the micro 3D printing. And I mean, that is quite exciting. But I think because I use the laser-based printers, those I like those the most. But, you know, all of them produce like really great parts for each different application. But I'm going to stick with mine because, you know, I like it more.
Okay, that's good. Yeah, we're not getting many questions from the audience, so please, I will remember people to just feel free to ask any questions in the comments, both on YouTube and Facebook. Just let us know what you want to ask our favorite speakers. So
yeah, I think as Amanda said as well, I'm on Twitter as well. So if anyone wants after to ask questions, they can feel free to do it at me, and I'll reply them without a problem.
Yeah, that would be great. I think it's good to have like follow-up conversation, and the now we're broadcasting live, but then the video will be available so people can get back to this later on. Okay. Also, I'd like to ask you what attracted you to SoapBox Science in the first place?
Yes, so I quite like to do outreach. I was doing a few outreach before, well, everything that happened this year. And I really like the idea of when it was, you know, in person, of just, you know, being in the middle of, I don't know, of the square and just talking about science and also women in science. I really like that they focus also on that, because I think we need to show that, you know, a lot of us can do science perfectly. It's not only a boys' club. So I really like that idea, and I like to interact with different people as well. So yeah, not necessarily colleagues or peers. I like to just make everyone aware of science. So that was quite nice.
Yeah, it's true. I think it's a great platform to reach other people and to showcase our science. Yeah, we have a question here. So someone asked, what will be the cost of personalized medicines using 3D printing? Do you think the costs will be too expensive or will come down and will be more?
So I think it will be like with every new technology. Maybe just right at the beginning, it might be a bit more expensive until, you know, it gets a bit more used and a lot of maybe hospitals or other things have the printers. But after that, I think it can be cheaper as well, because, for example, if a certain patient needs less dose, then you can put that dose in the printer, and you also do use less drugs and materials that you don't need to waste, so you're saving on that. You can also save time, you know, and some of the printers are quite easy to use, so you can leave it running, and then, I don't know, the healthcare professional can do other things. You're also saving on that side, saving on time. So I think it can get cheaper, but it might take a little bit of time until it gets more used. So I can't give you a specific quotation of price, but I think with time we will, you know, save money for sure.
Okay. Thank you, Alice. It was great to have you here today, this morning. So yeah, people can post. Oh, wait. We have another question. So they are asking if the drugs in this device could be injected to the eye, if it's currently in use, or it is previously not be possible to get these drugs into the eye at all.
Yeah, so the ones that I was focusing for my project, so these drugs can be injected in the eye, but the problem is that they need to be injected very frequently, once a month each, and the treatment can last for 2 years, which means you have a lot of injections per year into your eye. And the issue with this is that it causes many side effects in the eye. It can cause like cataracts, some hemorrhage, different side effects, because it's too aggressive to the eye to receive so many injections. So then the idea of the project would be for this implant to just be injected maybe once or twice a year. It will release the same amount of drug with 6 injections, but just or 12 in one go. So then you will have maybe half the amount or even less of injections. You will still get your treatment, but without all these side effects. So there's a few companies, like a couple of them in the world, that are trying to also do something similar, but not with 3D printing, because there really is a need for this type of treatment for patients. So that is the main point of it.
Okay, thank you. So I'll go to our next speaker, and so I will introduce Samantha Harrison. She's a PhD student at the Nottingham Biomedical Research Centre at the University of Nottingham, and the title of her talk is "Shining Light on the Brain: How Lasers Can Help Us See How We Hear Speech." So, totally different topic now. So let's go ahead, please.
Hi everyone. I'd just like to start by saying a big thank you to the SoapBox Science team for having me and for putting together and adapting this event so that we can all still discuss science even in these unusual times. My name is Sami and I'm a PhD researcher with the Nottingham Biomedical Research Centre and the University of Nottingham. My work is about how we hear speech, how we identify, process and understand spoken language, or I guess more specifically, why some people don't. To be able to process spoken words, you first need to be able to hear. Some people who are born deaf or become profoundly deaf can have their hearing sense restored by a prosthetic device called a cochlear implant. There are a number of things which affect how well a cochlear implant works for an individual, and one of the biggest things is age at implantation. The younger someone is when they receive their implant, the better their outcomes tend to be. Because of this, many babies who are born deaf receive cochlear implants very, very quickly, often before their first birthday. The majority of these children go on to have really great successes with their implants, but unfortunately, some do not. For those of you who have ever had a hearing test, you might recall being asked to press a button or move a toy when you hear a beep. Or maybe you were asked to repeat a sentence or identify a word. Now with babies, we can't just give them instructions and expect them to be able to do what we're asking. And I think we'd have a hard time trying to get them to repeat sentences back to us when they haven't even learned to talk yet. Because of this, babies can't complete the tests we typically use to assess how well a cochlear implant works for an individual. This means we don't really know how well they're doing or how much rehabilitation and support to give each child until they're a few years older. When we're really little, our brains develop and change very quickly in response to the world around us. As we start to grow, some of these changes are much harder to achieve. So if a baby is struggling with their implant for a few years, they might be struggling to understand and process speech, and this means they may never develop this ability to the same level as their friends who don't have hearing difficulties, or their friends who have good cochlear implant outcomes. Therefore, it's critical that we find a way to predict which babies might not have particularly good outcomes very early on, so that we can offer extra support before they pass the period where it's harder for them to adapt and develop this new hearing skill. Hopefully, this intervention can improve their long-term outcomes and be really useful. But how can you predict who might or might not be able to understand speech in a few years' time? Unfortunately, time travel and crystal balls are apparently very unreliable measures. So instead, we look to the brain. Research in adults has found that the way your brain responds to things in the environment around you might have the answers. In people with normal hearing, sounds and speech, such as me talking to you now, are processed mostly in the auditory areas of the brain, which is around here. If you watch something visual, such as me moving around, this is mostly processed in the visual brain areas, which are at the back of your head. In deaf individuals, though, this might not be the case. When these auditory regions of your brain aren't getting much or any input, your brain doesn't just sit there and do nothing. No, that's prime real estate right there. Instead, your brain finds a way to repurpose the area. One of the ways it can repurpose this area is to use it to process visual information. This is called cross-modal plasticity, or cross-modal takeover. This change in the brain might help explain why some people struggle with their implants, and might give us clues about which people will need extra help, or even give us a way to see whether or not our extra help is working. So how can we look at the brain to observe these changes? The most common form of brain imaging is called MRI, or magnetic resonance imaging. As the name suggests, this uses a big magnet which isn't great to put near someone who has a metallic and magnetic implant inside their head. MRI is also very noisy, which is not great if we're wanting to see how their brain responds to sounds in a quiet environment. EEG, or electroencephalography, is another common way of monitoring the brain, and it is much quieter. But the implant can interfere with the signals it records. So what method can we use? Well, there is a method called functional near-infrared spectroscopy, or FNIRS. FNIRS is very quiet. It's not affected by cochlear implants, and it's suitable for people of all ages, right from newborn. FNIRS uses beams of near-infrared light, which are basically harmless lasers. The participant wears a headset, and this headset sends these harmless lasers through the hair, scalp, and skull to the outermost layers of the brain. The blood there absorbs some of this light that we've sent and reflects the rest back, and the headset picks up how much of this light is returned. By measuring how much of this light returns back to the surface, we get an idea of how much blood is in the area of the brain, and we can monitor how this increases or decreases when we give some stimulation, such as a video to watch or a sound to listen to. And if the blood is increasing, we can assume that part of the brain is active and that it's working hard. So how does FNIRS fit into my research? Well, I'm using FNIRS on deaf babies to see how their brains respond to our different types of stimulation. I'm specifically looking at the auditory areas of their brain because I want to see that cross-modal plasticity that we talked about earlier, when the auditory areas get used to process visual information. And I want to see how this changes once a child starts using their implants. Then we can look at all of this information and see if any parts of it, any patterns or any changes, can predict which children will go on to do really well and which children might struggle and need extra help. My research got halted quite abruptly, thanks to coronavirus. What we have so far has shown that FNIRS works really well on normally hearing children, and it picks up brain activity in response to the videos and sounds that we've developed and chosen to play. We know from previous adult research that information from FNIRS measurements can be predictive of cochlear implant outcomes, so we hope to be able to show similar in our babies very soon. In the meantime, we've been running surveys, both for parents of children with cochlear implants and the clinical staff who work with them. We want to try and find out what sort of things they would or would not like to be included in an FNIRS-based tool. After all, there's no point in developing a tool for clinics if we don't develop something that they'll want to use. Hopefully, as the research in this area progresses, we'll be able to use FNIRS in hospitals and clinics to predict and measure cochlear implant outcomes, especially in babies and young children, as they can't use the other methods we currently have available. If we do that, we will quite literally be shining light on the brain and using lasers to see how we hear speech. Thank you.
Yeah, and thank you, Sami, for your presentation. I will remind our audience, please ask questions in the chat. We are reading your questions and we can ask them live to all our presenters. So, Sami, good morning. How are you today? I'm good, thank you. So thank you so much for participating in this session. So we have a few questions already from the audience. People are engaging more now. So there's a question regarding because your work involves studying children, have you found that COVID has changed your research, and how will it change it going forward?
Studying children didn't affect the effect of COVID didn't affect that so much, but obviously, as I mentioned, the research completely got halted. We're just starting back up now. Obviously, we haven't been in the lab since March, so it's a steep learning curve getting back to that. But we've been trying to find other forms of research that we can do that will still help drive this forwards. That can be done online, so like the surveys we mentioned, finding out what parents and clinicians do and don't want us to be looking at. So we don't want to be wasting our time or the research funds if we're going the complete wrong direction. But we're starting to go back into the labs, like I said, very soon, learning all the new PPE rules that we have to follow for that, so it's been quite interesting. But overall, hopefully, not too much of an impact other than this big time delay.
Okay, and how well do the babies respond to the hat in general?
That is very, very mixed. Some children absolutely love it, especially if they're kind of kids where their parents have said, oh, you're going to wear a really special hat today, and they come in really excited and where's my hat? And there are others who will scream and cry and really don't want it on. Usually once we've distracted them with a few toys and we've got some videos of penguins and things they can watch, they tend to settle right in. Once it's on, you can't tell. It's just the process of all the adjustments that we have to do. It can sometimes be a bit tricky.
Okay. We also have a question. How deep can you detect, and if it's deep enough for a good result? So I guess it's about the penetration of the laser.
Yep. We can detect down sort of an outer cortex level, so the very outer area of the brain. For what we do, that is deep enough, because the sort of auditory association areas we look at are right out on quite a big bump, which is nice. But if we wanted to look at something that was further into the brain or down into some of the dips that we have, we might struggle a bit. It largely depends on each participant, how thick your scalp and your skull is, how close to the surface your brain is. But for the most part, we would just look at the very outer surface. If you wanted to go deeper, you'd probably have to start using something like MRI and consider using a different patient population.
Okay, that's good. Yeah, just a general question. What's the most exciting experiment you have ever done?
Probably this one, just because I get to work with so many different people, and obviously working with babies brings complete different challenges every single time. But I love all the research I've done, especially any that involves brain imaging or monitoring blood flow. So don't really want to pick a favorite, but it would have to be this.
Okay. And if you had all the money and time in the world, what experiment would you like to be able to do?
We would love to take this one, but onto a much bigger scale, and look at a huge cohort of cochlear implant children of different ages and really track them from possibly even birth, if they're deaf from birth, and see how they change and adapt right up to teenagehood. That would be great, because at the moment we're very restricted to a short time frame and just a small number of children.
Okay. Yeah, I understand that. Okay, thank you very much. I think we're done with you. Well, I will now introduce our next speaker. That's Gamze Presentation. She's a PhD student at the University of Nottingham, and her talk is about the use of yoga in the management of blood pressure.
Hi everyone, I'm Gamze Presentation, a second year PhD student in the Division of Epidemiology and Public Health at the University of Nottingham. Today I'm going to talk about my research project, which is about the role of yoga in the management of hypertension. Firstly, I will explain what hypertension is and what causes hypertension, why it is important to manage hypertension, and then I will talk about what I do as a research. Firstly, our heart contracts between 60 and 90 times a minute. With each contraction, our heart exports blood to our arteries, and the blood exerts a force to our arteries when it travels. So, the force applied by the blood when our heart contracts is much stronger, and this is called as systolic blood pressure. When our heart is relaxed, the force exerted by the blood is less powerful, and this is called as diastolic blood pressure. If this systolic and diastolic blood pressure persistently increases, people are diagnosed with high blood pressure or hypertension. So, what causes this increase in the blood pressure? There are several factors that might affect the increase. And these can be divided into two groups as non-modifiable risk factors and modifiable risk factors. So, as we can understand from its name, non-modifiable risk factors, those are that we cannot change. These include age, ethnicity, family history, and genetic factors. So non-modifiable risk factors are those that we can change, and they are a part of our lifestyle. So luckily we can change them. These include unhealthy diet, physical inactivity, smoking, excessive alcohol consumption, and persistent exposure to stress. So, why it is important to manage hypertension? Hypertension is linked with serious health conditions like cardiovascular diseases, heart failure, stroke, chronic kidney disease. And the population in the world having hypertension is really huge. It's estimated that around 1 billion people have hypertension. So hypertension related health conditions are also likely to increase because of hypertension. And also the cost of hypertension and hypertension related health conditions is expected to be nearly 1 trillion US dollar. So it's really important to manage hypertension for individuals having it and for health systems. So here comes to play yoga. So what is yoga? Let me explain what is yoga. Yoga is mainly accepted as a part of complementary and alternative medicine, and it's a practice as a kind of exercise, health practice, and relaxation method. So yoga is actually originated in Indian subcontinent around 5,000 years ago. And yoga as a word means to bring or to unite. So it aims to unite the mind, body, and spirit. And yoga is becoming very popular around the world. It's being accepted by the people. So there's a potential in yoga to help those people. So what I do as a research, I do a systematic review to understand if yoga really is effective to manage hypertension and what aspects of yoga are more beneficial. So what I do is I bring together what other researchers have done previously and to get a more robust and generalizable results about the effects of yoga on hypertension. So then what I do is I do interviews and survey with yoga teachers and therapists in the UK to understand the potential of yoga in the management of hypertension in the UK. So also the systematic review will help to understand if the potentially eligible yoga practices are available in the UK and who can access these practices. So this will actually help to understand if really there is a potential in the UK to use yoga as a way to help hypertensive people. So what yoga teachers and therapists think about this? What are their experiences and how frequently they confront with hypertensive students in their classes? So this will help to understand the potential of yoga as a way of managing hypertension in the UK. So that's what I do. Thank you very much for listening.
Yes, thank you. Let's welcome Gamze. And please don't forget to post your questions in the chat below. So both on YouTube and Facebook Live, you can ask direct questions to our speakers today. Hi Gamze, how are you today? I don't think we cannot hear you. No, I don't have any sound from you. No, sorry, I don't think. Yeah. Yeah, we'll try to sort out this technical problem. So please don't forget to ask any questions. This is a very interesting topic on yoga and its effects on our high blood pressure. So please, please try to get in touch with us and let us know what you want to know about this. I think this is one of the problems people now are getting used to with all the Zoom and Skype and Teams meetings around. So everyone has problems muting or with sound or video or anything. So, and also unexpected interruptions. So we have to deal with this technical problem because we're doing live and things happen. So just keep up with us, and we want to know the questions you have. Yeah, I see people are coming, are starting to put things.
Okay, so I will write from chat.
Do we have sound from Gamze already? Yes. Hi, Gamze.
Can you hear me? Hi.
Yes, finally we can hear you. Yeah. So thank you for keeping
Sorry
up
for
with us.
the
No worry, it happens all the time. It's just technology. So we have a question from the audience. So they ask, how often do you have to practice yoga to help control blood pressure?
So actually, according to my systematic review results, there are quite heterogeneous studies about this. So I can't exactly say how often you need to practice because it changed actually according to the type of yoga you are practicing. And so mostly breathing practices and yoga poses actually are helping, and meditation if they are practiced together. So I can say that there are some studies doing only one-time yoga session, but it's not well enough to reduce blood pressure in just one session.
Okay.
It needs actually a regular practice. I just can say that.
Okay. And the people are asking if is yoga effective for other diseases as well, or that's not what you're studying?
Yeah, there are really many research ongoing about yoga and health conditions. So there are many research about rheumatoid arthritis and yoga, back pain and depression and diabetes, many actually. I'm just working on one of these health conditions that yoga can help.
Okay. Yeah, I practice yoga myself, and I know that it helps me feel better, but I never quantified how effective it is to reduce any health condition. So do you have like any differentiation between different types of yoga? Because there are so many different, if you go to a gym, you can see there are so many different types of yoga. Is there any difference on how they affect you, or basically it's all the same?
Actually, again, I will talk about the research I do. For now, it looks really difficult to say just one style works, because actually, if we think about yoga as a general, almost all of them come from one style. It's generally called as hatha yoga, and then all styles vary. Now there are plenty of different types of yoga, and it's difficult to say just one of them are working. I can say that different styles use different props or different practices, but mostly the main idea of yoga is to just be on the moment, at the moment, and not thinking about the future of task and just feel yourself and try to be relaxed.
Okay. Yeah. So there's a question of what is actually happening during yoga. Is there a release of certain chemicals in the body?
Yeah, yoga actually, this is again not approved. Sorry, this is not exactly evidence. Sorry, I couldn't find the word. There is some research suggesting that yoga can help to increase the parasympathetic nervous system activity. So this will actually help to manage hypertension. And this is one of the ways of helping to hypertension through yoga. And the other method, except these hormonal activation, and there are other aspects like physical aspects of yoga and stress relieving aspect of yoga. So sympathetic nervous system actually is mostly causing the tension, stress on the body, and yoga helps to reduce the activation of this system.
Okay. So there's another question. So you mentioned that there are many studies about the influence of yoga on hypertension. So do you consider this is a popular area of research?
Yeah, I think it's really getting more attention because, especially about hypertension, it's very common health condition in the UK and in the world. And I can see that from the research, people have more tendency to seek complementary therapies because mostly elderly patients are hypertensive people and they need to use combination of medication, and this makes a little bit tendency to seek other treatment methods. And that's why I think yoga is getting more attention because it helps people to feel more relaxed and it helps for their blood pressure as well.
Okay. So I just a general question. So when did you realize that you wanted to be a scientist?
I think when I go to the Netherlands for my Erasmus project, I realized that I wanted to really work on academia and need to learn something that I can really research about. Because I thought just practicing in the hospital or medical aspect will not satisfy me. I know it's very good thing to do, but I thought maybe I can help more than doing this. So if I can search something that I really like, I can help people.
Okay, thank you so much, Gamze. Thank you for being with us today in SoapBox Science. So we're moving forward to a new speaker, but don't forget to ask questions in the chat, both on YouTube and on Facebook. We are reading them, so just interact with us and with our scientists. So the next speaker is Dr. Jihan Al-Jawari. She's a lecturer of biosciences at Nottingham Trent University. And the title of her talk is How Stem Cells Can Help Resolve Problems of Bone Healing. So let's go ahead with her video.
Hello, my name is Jihan, and I'm glad today to have the opportunity to talk about my research in the area of the bones and stem cells. So bones, or the skeleton, is very important for our life, not only for movement, but also for other metabolic functions. Stem cells are very specialized and unique cells, which naturally can form into bone when there is a damage or a fracture. Luckily, bone healing occurs normally and without scar tissue in many cases. However, because of increased life expectancy and increased activity or life activities and exercises, accidents can happen. The definition of complicated healing or delayed healing as simply means when there is more than 9 months after the injury date without healing. And this is quite problematic for many reasons. First, it means long absence from work or study can lead to mental issues, and obviously social problems lead to an increase of a need of care, in addition to health burden, because the patients would need more frequent outpatient visits to the hospital, and eventually these patients will need surgical interference. In most cases, stem cells now are considered as a therapy when they locally added into the injury site. So how this can happen? I will show you here a photo of the pelvic bone for a patient, and this is the process of aspiration or taking a bone marrow samples. And the bone marrow, as you see, it's a blood-like structure, a liquid, which contains a sort of similar cells to the blood, but something special, which is the stem cells. And usually this bone marrow can be a very precious part to be used for therapy of complicated healing. Although this type of therapy is quite at early stages and not a routine, it needs optimization, and this is where my research lies. So in my research, we're trying to quantify or count the numbers exactly of the stem cells in the bone marrow samples, because the fact is, interestingly, these numbers are very variable between different donors. They also can be changed in the same donor with aging and maybe with other factors related to their health conditions, medication, and so on. My research also is looking to the deep understanding of the bone healing process. And this is a very complicated process because it involves interaction and collaboration of different cells. So stem cell, which can form bone, usually needs help from other surrounding cells. Most important ones are immune cells, and also the cells which line the blood vessels. Another part of my research is about using materials, and let me show you what I mean. These are sort of biological materials, a 3D structure or three-dimensional materials, or we call it scaffold. This structure also can be used in stem cell therapy to support and hold the stem cell, which we apply to a site where a fracture is, or a damage, a big damage of a bone. And my research is looking into how the stem cell behave on these structures and what is the best material to support the healing function of the stem cells. So as I say, the main goal or main research to improve the therapy for the healing, considering different factors. Part of it are cells or biology, and part of it is also looking into the physical materials. We hope by optimizing this kind of cell therapy to interfere as early as possible, and in the cases which we anticipate complication will happen, to save effort, to save any health issues, social issues, mental issues for both patients and his health system. And most of all, our target is to improve the quality of our life and never stop moving. Thank you very much for listening, and I will be happy to receive any question about my research. Thanks.
So don't forget to ask any questions about this talk in the chat. So you can do it both on YouTube and Facebook. We are happy to answer any of your questions. So we have some questions already here for Jihan. Hi, Jihan.
Hi, how
this
are you? Yeah,
morning.
I'm lovely to be with you. Thank you for the invitation. Thanks.
Yeah, thank you for being with us. It's great to know a little bit more about your research. So there's a question about stem cell therapy. Is it already widely
Indeed,
available?
they are widely available because they can be taken from the patient tissue, so or what we call it autologous. But in terms of a real application in the clinic, they mostly limited. So the resource is really available, but because the studies are still ongoing, comparing different source or type of the stem cells. Some centers, for example, focus on bone marrow source of the stem cells. Others would prefer looking into stem cells derived from adipose tissue. But yeah, in general, it's very worldwide concept of therapy.
Okay, so can you give us some examples of diseases that are already being treated with this therapy?
Yeah, so in terms of healing, bone application is the one field, and it's my field of interest, but also stem cells can be used for cartilage repair, and that's for diseases like osteoarthritis, where the cartilage of the knee or ankle damaged. So also an interesting thing for mesenchymal stem cells, or these like bone marrow stem cells, can be used is for some disease where the immune system is also hyperactivated, which we call this group of diseases autoimmune diseases, for example, systemic lupus, Crohn's disease. So again, there are a lot of clinical trials about using these stem cells because they have other sides of function in addition to the healing, which is control of the immune system. So really, there is a big potential for using these cells in almost majority of diseases, really.
Okay, so at which stage of bone healing would you consider to using stem cell therapy?
Yeah, that's a good question. Well, there are three stages of the bone healing, really. First stage or early stage, where the inflammation and immune cells are attracted to the site of injury because of the damage happening. And this is the stage where we expect the interaction between mesenchymal stem cell to recruit and start interacting. And then while immune cells response in finished or complete, the stem cell will still play an important role in the second phase, which is the healing of the bone marrow. And then once they transformed into bone cells, the role will finish at that point, and we will reach the third stage, which just remodeling or reshaping of the healed, newly formed bone.
Okay, and can you use this stem cell therapy at any age, or?
Yeah, that's a great question. Well, unfortunately, the complication usually happens after age 50s. And this is why we focus on our research into the look of the characteristics and function of these stem cells in this age group in particular, because we expect this is the age where will be most really beneficial. The stem cells will be more beneficial for this age group. We have very good preliminary, very good results that the stem cell in this age group is functioning well, but their number could be in general less than the young patients. And this is something the science is looking forward to sort of enrich this number or improve the quality of even the few numbers by stimulating these cells to act at a higher level.
Okay, thank you. So there's another question. What excites you more about working with stem cells?
I think I'm very excited first because it's that kind of therapy which is, as I say, it's a natural therapy. It can derived from the patient or a human own body. So no synthetic addition, although there is another line of stem cell going on the world as well, which is taking stem cell from donors and expand them in the labs to produce very, very large number of cells, which is also useful. But as I say, my research is more on the line of using sort of native cells or natural cells, which can be extracted from the patient and relocated from bone marrow and relocated at the injury site with minimum manipulation or processing. And the advantage of this is to avoid any immune reaction or any risk of infection being from other source or being processed in the lab. Also no possibility for any genetic or any changes. So really, it's just this therapy would need a little bit of optimization in terms of the doses and in terms of how to enrich the numbers. But I think I can see a lot of potential and advantage in this field.
Okay, so you see it as a medicine of the future and a solution for a lot of...
Yes, definitely, yeah.
Okay, thank you so much, Jihan, for being with us today. I will now introduce another speaker. So it's Dr. Rachel Bolton-King. She is Associated Professor of Forensic Science at Staffordshire University. And her talk is about investigating shooting incidents. Can we rely on humans?
Hi, my name is Dr. Rachel Bolton-King, and I'm an Associate Professor of Forensic Science at Staffordshire University. My particular area of expertise is firearms and forensic ballistics. And as a result, I'm here today to talk to you about investigating shooting incidents. And can we really rely on humans? Well, it's a really complicated question because investigating shooting incidents is actually quite a complex situation. It involves lots of sectors and disciplines working together, and lots of professionals are involved in the investigation of shootings. And shootings not just being where people are hurt or harmed, but also things like criminal damage or things where firearms might be discharged as celebratory fire, or a wide variety of firearm-related offences that don't involve physical shootings. So if we do consider just shootings, then you're actually looking at a whole range of different professions that have to work together within the criminal justice system in order to ensure that we deliver justice to society. And as a result, we could be talking about working with communities, talking about working with police officers, crime scene investigators, forensic scientists or forensic ballistics experts that work in the lab. Also, we could be considering the prosecution, so the lawyers, but also the defence lawyers as well. So looking at both sides of potential convictions. And as a result, it's really, really hard for all of these sectors to work together. But humans are the fundamental basis in which any kind of crime can be investigated or predicted or prevented and subsequently prosecuted against. And humans can be quite a big challenge and a big barrier to implementing change and for things to be working as effectively and as efficiently as we might like. And as a result, part of the work that I do is around looking at working with police forces, working with our national bodies such as the National Ballistics Intelligence Service, and looking at how we can improve the workflow and what happens from the start of an incident being detected or a crime being committed, right the way through to ensuring that the right person is prosecuted. And therefore, it's really vital that all of the sectors, we consider the entire chain, but it's really easy for us as humans to compartmentalize and to work only within our particular discipline. So for example, the forensic lab or the crime scene or as a police officer. And so the research I've been currently doing really needs us to better understand everybody's role and responsibility, and to be more aware of actually what firearm offences are, what types of firearm evidence there could be at a scene or in a search when we go to someone's home or workplace, etc. Identifying illegal manufacturing, for example, that might be going on with firearms or ammunition. But even detecting and reporting that a crime has happened, there's challenges that us as humans actually face in order to do that. You know, that might be fear, or it might even be just accidentally or unintentionally not realizing that what we hear as gunfire might be, we might think it's a car backfiring or fireworks or something else, and therefore we don't actually report it. So research has been done internationally looking at how often people report crime to emergency services such as the police, and actually 80% of the firearm discharges never actually get reported. And as a result, there's this massive amount of forensic evidence that's not actually gathered because we don't even know a crime's occurred. So no one goes and collects any evidence. And as a result, there are lots of crimes that might be happening that we don't know of and that we're not linking together. But we need to actually use technology in order to help us link these crimes together, and also to help us identify patterns or threats that might be emerging within our communities or in our region or even nationally. And as a result, there's lots of work that's ongoing in order to try and develop these technologies that enable us to link crimes together or identify these patterns and trends. So analysing the crime data that we do know about. So in terms of detecting firearms discharges, so shootings, we can utilise more adaptive systems like gunshot detection. And this is, for example, ShotSpotter, you might have heard of. They're heavily used in the United States and South Africa. And this allows us to objectively monitor an environment to detect if there's any gunshots that occur that we might not know about, and then deploy police rapidly to the scene. That is great in terms of making sure the ambulance are called and that people may not die and more likely to survive, but also it's helpful in terms of gathering that forensic evidence. So what is forensic evidence in terms of firearms and ballistics? What does it look like? Well, it could look like this, really, really big. Could look a little bit smaller, like this. Or it would be really, really teeny and look like this. They're fired cartridge cases. Alternatively, ammunition, you might have been seen that looks more like this. So you've got a projectile attached to that cartridge case. Or we have courtroom aids as well, so that you can actually look at the projectile and the fired cartridge case on a much bigger scale. And in terms of that the image that you can see on the screen here. So this is a fired cartridge case that has been imaged with three-dimensional technology. Such as the Evo Finder. There are other systems out there, but we're doing research to determine what are the factors that affect how well we can link one fired cartridge case that's been discharged from the same firearm to another. So things like ammunition materials, do they affect the tool marks? Yes, they do. But how much do they affect it? Does it stop us from linking them together? Generally, no, it doesn't, which is fantastic. but also helping us to use machine learning in order to help us more objectively determine what is similar or what might be different between fired cartridge cases from the same gun versus from a different gun. And so some of the work that I'm involved in is looking at collecting big scale amounts of data in order to differentiate and support firearm examiners in order to make those connections or make those eliminations. And as a result, this objectivity is really important, because we can make mistakes. We are human. And unfortunately, there have been mistakes that have been made, and we want to rectify those moving forward. And so we can use these more objective systems in order to help us as humans, and therefore we can help minimize the chance of miscarriages of justice moving forwards. But also we can hopefully link more and more crimes together that we might have missed as humans, which we're only able to look at or consider relatively small numbers of cases at a time and process them. So these databases and this new technology, it's not new in the sense it's like just come out. It's been around for 20 years or so, but we're using it much more effectively and much more informatively in order to support our investigations and to help us as humans. And also using statistics, so calculating likelihood ratios, for example, can help us evaluate the strength of the evidence. How close is that connection between one case that's been fired to another case that's been fired, and have they been fired from the same firearm or from a different one? So we consider different hypotheses and alternatives. And so when it comes to can we rely on humans, mostly the research shows that our subjective opinions, our human opinions, are sometimes more accurate and often more accurate at determining the right interpretation than computers can be at the moment. But as technology and as computer algorithms advance, as we do more research and we know more, then there's a possibility that they might be able to be just as good, or in fact exceed what we can currently do as humans. But at the moment, yes, we still need humans, and I always foresee us still being able to and need to use humans. But it's about how do we generate the right practice, have the right policies, use the right technology to really help maximize the amount of evidence that we can collect and the intelligence that we can gather, and help us to be much more timely and effective and efficient in producing the right criminal justice outcomes for everyone in society. Thank you very much for listening. I hope you found it of interest, and I look forward to any questions that you have.
Thank you very much, Rachel, for this exciting work and for being with us today. It's a really lovely area of research, and it's quite important as well. So I'm just looking in the chat to see if there are any questions for you. One of the questions is asking about, do you think artificial intelligence could be useful in this area?
Yes, and when I mentioned machine learning, absolutely, this is where algorithms are looking at the data. So it could be a three-dimensional image of a fired cartridge case or a fired bullet, and looking to see what are the features that really differentiate the cartridge cases or the bullets that are fired from different weapons, to help us to really look at really big databases that are full of thousands and thousands of similar items, to basically rank them to see, whether there are any others that are already in the database, other crimes that have happened that we can link together. And absolutely, artificial intelligence is already being utilized in order to help us achieve that end outcome, and support examiners to identify in a really timely way if there are any other cases locally or nationally, and sometimes even internationally, that police officers need to know about to strengthen their investigations and to find more leads to help them pursue and solve, hopefully, a crime.
Great. Seems like it's a promising area. Another question is asking about are there any examples or comparison study where they are comparing the a human bias compared to artificial intelligence or computerized system?
In terms of the artificial intelligence, I don't think so at the moment, but there have been in terms of whether the algorithms that are being employed by ballistics imaging systems, whether they can what their error rates are, and comparing them to humans, their own comparisons, their expert opinion. And it has demonstrated that the experts, do more often get it right, and they make the right interpretation and conclusion from the research that's been done. But it's also demonstrating that the algorithms, the computational algorithms that are now being employed, are also as effective at being able to identify that there are other linked and associated items within the database, and they can correctly list basically those that are closest to it in that first rank position, that closest rank position, just as well. So it's not necessarily at the moment that artificial intelligence and technology is going to be used instead of humans. It's about working together in order to use the technology to support the expert's opinion, and hopefully we will continue to move forward to make those objective connections.
Brilliant. I have more general questions for you. Is this your first outreach event, or have you participated before in other activities?
I've definitely participated in other outreach activities before, but this is my first Soapbox Science event.
Brilliant. Well, it's really lovely to have you here.
Thank
so
you.
when did you realize that you want to specialize in this area?
Well, I've always been really fascinated by forensic science. So even before crime scene investigation or the CSI program even became a big television program, I've always wanted to do it from a really young age. I think I was about 7 or 8 when I realized that I've always wanted to use science to help solve and investigate crime. But firearms and ballistics as a subject, I certainly wasn't interested in at all until I was at university. So I did my undergraduate degree in forensic science at Nottingham University, and it was then that I did kind of a module in firearms and ballistics, and I really kind of found it really exciting. And I've always found it hard to be able to pick biology or chemistry or physics. And this particular discipline is so diverse. I just love it. And it requires me to have a broad understanding of all those disciplines. And it means that I conduct research across the disciplines, and often it's sort of using multiple disciplines all together, including kind of the criminal justice aspect. And it was really then that I realized that I really wanted to focus in this field rather than sort of drugs analysis. And that's when I finished my undergraduate degree, I thought, you know what, I don't want to stop learning. I don't want to stop researching. I really loved research in my undergraduate degree, and I decided to do my PhD. And this was a fantastic opportunity, again at Nottingham University. And I was able to pursue ballistics imaging and firearm identification in that. And that's exactly, when I realized that, the role of and the job of academia was for me.
Oh, brilliant. It sounds like a really exciting journey.
Definitely.
So is it difficult to take your path being a woman?
I actually think for me, it's possibly been a bit easier because I'm a woman. When I first started in the firearms and ballistics area, it was heavily dominated by males. And over the last 10 or 11 years, it's been amazing to see the sheer number of female firearm examiners start being trained and start to pursue their field and their career in firearms identification and the field of firearms and ballistics. And I think that's just fantastic. And I'm really glad to be part of that change and that wave, and to see the number of women sort of in that sector. But the other thing I found that, you know, the firearms and ballistics field of forensic science was so much more open, so much more collaborative, so much more supportive than I found sort of other areas in the past. And so, you know, actually, I think being specifically a young female at the time, when I finished, when I was doing my PhD, I think really helped to progress my career. And I've been really fortunate to work with some amazing people across the world who have really supported me, and both in practice, you know, in police forces and in forensic laboratories, etc., as well as universities. And I couldn't do what I do without working in that collaborative environment and that supportive environment. It's just amazing.
Oh, that's really encouraging to hear, and hopefully it will inspire other young girls to take your path. We have another question from the audience asking about what do you think about regarding the frequency of gun crime reporting? Do you think there is a link between the frequency of gun crime in a particular area and the frequency of gun crime reporting? Or in another word, do you think that if people live in an area where there is a lot of crimes going on, will they become desensitized and then therefore they don't always report what they see?
Yeah, it's a really interesting question, and something that I looked at in my Churchill Fellowship. So I won a Winston Churchill traveling fellowship last year, and I went to South Africa and the USA and France. And I think this is really complex, and sometimes in high gun crime areas, it might be that those people living there are really quite fearful of reporting gunfire, or it might be that that is unfortunately their normal, that's their normal day. Hearing gunshots and having firearm related activity is part of their daily life, and therefore it would happen so often that they have become desensitized to it to some extent. We do, even within a region though, we do find that those areas that are slightly more affluent, where gun crime may be slightly lower, and people may be less fearful and more likely to report that crime. But I do think that gun crime occurs regardless of where you are. We just don't necessarily know about it, and it's only the ones that we hear, like the actual physical sounds of gunshots, that we recognize as being so, that enable us to report. There's a lot of other firearm offences that people don't think about, you know, and I mentioned it in my talk as well, around illegal manufacturing of both firearms and ammunition, converting those firearms that are maybe starting pistols and converting them to fire live ammunition. And so there are a whole range of firearm offences that people don't necessarily think about, and they don't have an audible sound to detect, or there's not directly a victim that you can see that, you know, is hurt and therefore alerts you to the fact that a crime may have occurred. It often starts much further down the chain. It may be the way in which firearms and ammunition are even trafficked and smuggled across borders, that, you know, initially you don't think there is a victim, but ultimately, without that trafficking, without that smuggling, and without that manufacture or assembly, there's never going to be a victim at the end. So there are lots of types of victim throughout that illicit activity that people often don't think about, and that's part of the recording and the reporting as well.
Yeah, it's quite interesting to see how diverse it is. Well, thank you very much for being with us today, and wish you all the best for your future.
Thank you.
So now we are moving to our next speaker, Dr. Hanan Ali Bosetta. She's a lecturer in pharmaceutical nanoscience at the University of Birmingham, and her talk today is about fantastic nano voyage.
Hello, my name is Hanan Ali Boussata. I am a lecturer and group leader at the School of Pharmacy, University of Birmingham. Today I'm going to talk to you about the future of cancer therapy with nanorockets. I'm sure you've heard the word cancer, or someone very close was diagnosed with cancer. It's a disease that is spreading quite vastly and quite rapidly, and it's really predicted already that there will be 27.5 million new cancer cases worldwide each year by 2040. We're talking about 20 years down the line, so it's not really far off. And this is really a massive increase of 61.7% from 2008. Therefore, there is an urgent need to quickly develop new cancer therapies. Now, do we start the wheel and make new molecules from scratch that will take a long time for them to be developed and be approved and out in the clinic? Or shall we repurpose current anti-cancer therapies? Current anti-cancer drugs are actually very potent. They're very, very toxic to cancer cells, but unfortunately, they're also toxic to normal cells, causing side effects like stomach problems, hair loss that cancer patients suffer from. Cancer chemotherapeutics has also have a problem, a really major problem known as resistance. So after a cycle of chemotherapeutic, the cells become really clever and can actually recognize the drug, and it starts pushing it out so it doesn't come inside the cell and kill it. And that's what we know of as resistance. So it's very important that the patients will have to receive another type of chemotherapeutic drug that the cell has not really kind of developed resistance against yet, and they might have to go into another one and another one, into cycles. So it's very important that we overcome this problem of resistance. Now, what we're trying to achieve in my lab is basically deliver current potent anti-cancer drug more specifically, okay, to cancer cells and to reduce their resistance. How do we actually do that, and how do we achieve that? Is by delivering those existing potent anti-cancer drug in a nano-sized delivery vector. So we're talking about a vector that is really, really small. We're talking about 10 to the minus 9 sort of a meter. We're talking about something that's even smaller and thinner than a human hair. By delivering the anti-cancer drug inside the nanovector, what we're trying to do is hide the drug inside the drug delivery system so the body cannot recognize it, but allow it to navigate specifically and peacefully inside the cancer cells. So we're masking the drug so the body can't see it, but also hopefully we'll overcome the problem of resistance. These cancer cells, as I said, they're very, very clever. So now what I want you to think about is this rocket as my nano-sized delivery vector, and the astronaut inside as my drug. Imagine that they are so small, as I said, in the nanometer range, so they're nano rockets. They're so small to navigate inside the body and are well tailored to reach the cancer cells, but not the normal cells. Imagine the face of a cancer patient who will now not only enjoy a long life expectancy, a longer life with their beloved one, but they will also suffer from no side effect, no problems like hair loss, stomach problem. They can go back into society and enjoy their life as they recover. Imagine a nano rocket having a fantastic voyage in the body and choosing which cell to lend into. In the case of cancer, that will be a cancer cell. This might sound like a dream, but will soon become a reality with the work that with the research that we're developing at the Nanomedicine Drug Delivery and Nanomedicine Lab at the University of Birmingham School of Pharmacy. Thank you for listening.
Thank you very much, Hanan, for your lovely talk.
Hi, Zahra.
And thank you very much for being with us today. It's really exciting to hear about your work and what you are doing.
Thanks, Zahra. It's a pleasure for me to be here today and to speak in SoapBox Science as well. Thank you.
Thank you very much. So we would like to know more about your area of research. So as cancer is the only treatment options that you can treat with these nanovectors?
Thanks, Zahra. That's an interesting question. So cancer is one of the topics that nanomedicine or drug delivery have worked on for a while. So we already have some nanomedicines that are out in the clinic, specifically in cancer therapy. We can also right now, actually, with the COVID pandemic, see that some of the the leading vaccine, like the Moderna and the Pfizer, are actually containing some nanoparticles or lipid nanoparticle. So it isn't really only cancer. Cancer has always been the one topic that drug delivery have kind of worked on, or nanomedicine have kind of approached, but we can actually do quite a lot more with nanomedicine. As I said, the COVID, we're also doing some work on developing some nanoparticles that could actually cross the blood-brain barrier to deliver therapeutic to the brain for neurodegenerative disorders, for example. So I think cancer is just one of the topics that nanomedicine can play a role in.
Well, that's really exciting to see that the field is emerging and there are more applications coming on. There is one question from the audience asking that you have tried to mimic this the delivery vectors that you design with the rocket. So they're actually asking what happens to the rockets after they have done their job.
So depends what nanomaterial you're working with. Some of them are biodegradable, so they can actually deliver the therapeutic molecule and then disintegrate eventually. Some of them are still a kind of basically biodegradable. We're looking into how they degrade. So some materials can actually depends on what you actually do to it, how you actually prepare it. They might be more prone to degradation. Some of the things that we do is actually looking into the degradation of those nanoparticles and also their toxicity. So what we try to do is develop those, what we let's say call them the nanorockets, develop them to actually safely deliver the drug and eventually kind of leave the body, either biodegrade or kind of get excreted either via the urine or other kind of clearance mechanisms.
Mm-hmm. That's really exciting. Another question here. People are asking, how do you fabricate, design, or engineer such vectors to target specific cancer cells and not other cells?
Right, very, very great question. So if we're talking about cancer in particular, there are, we take into consideration, so designing the nanovector, we need to take into consideration disease. So we know, for example, for cancer, there are certain, the lymphatic drainage is basically kind of low in the tumor. We also know that the fenestra has a particular size, and we then try to design our nanosystem to be well defined to actually get through and stay within the tumor. So what we use in there is what we know as passive targeting. So we're not really adding anything, we're using more the properties of the cancer, but also we can also use what we know as targeted or targeted delivery or active targeting. So we know that cancer cells, quite a lot of cancer cells actually have certain overexpress, or so they present certain receptor, but they're only present, for example, in the cancer cell and to a lesser extent in the normal cell. So when you design your drug delivery system, you can actually attach a molecule to their surface that is really kind of specific and can only basically target those receptors and not really get into the cancer cell. So in that way, you can then achieve specificity and target it, because your drug delivery system has that particular specific targeting on the surface, and they can only go and basically get into the cancer cells, but won't be able to get into a normal cell, for example, because it doesn't have those protein on the surface.
Oh, brilliant. Another question people are asking, can you briefly describe the methods that could be used to reduce resistance, and how does your work can help in this?
So one of the way, as I described in the video, the cancer cells become really kind of clever. So they kind of recognize the molecule. So if I take my astronaut as the example of the molecule, so if the molecule is getting into the cancer cells after a certain period of treatment, the cell starts pumping it out. It starts recognizing, let's say, the face of the astronaut. However, when you're trying to, what we're trying to do with nanomedicine drug delivery is kind of hide that astronaut within the system, that the cell now can no longer see it as the astronaut anymore. It will see, will basically see this drug delivery system, which will have a capability to get and enter the cell and basically then release the drug inside. So in a way, that's a way of overcoming that problem of resistance, masking the drug. The cell is no longer being able to see my drug or my astronaut.
Interesting. You mentioned the word also toxicity. So being like very small molecules that are not, I would imagine that you can't really see them by eye. How can you control the toxicity, or if there is any harm to the environment or also to the body in this case?
Thank you, Zahra. We do quite a lot what we call as nanotoxicology studies. So what we're trying to do is, when we're designing our drug delivery system, we want to make sure that the drug delivery system or the nano system here is non-toxic. So when we're doing, so what we tend to do with it is change a bit of the chemistry, add certain functional group, reduce its size, for example, as well. So play a lot with what we know as physical chemical properties. And at the same time, we're looking and studying whether there is any toxicity, how we alleviate it. So then we're designing some safe nanosystem before we even put any drug within them, basically. So in that sense, the drug delivery system will be non-toxic, and the only molecule, if we're talking about cancer here, an anti-cancer drug, the toxicity will only then come from the anti-cancer drug.
Thank you very much. I have more general questions for you. So what advice can you give to a young girl that want to take your path or do the kind of research that you do?
I would say follow your dreams, and basically the sky is wide, the sea is so wide. So remember that whatever you see, I mean, believe in yourself, I would say. What you can see other people do, you can definitely do it yourself. And that's something I always say to my students. Believe in yourself and do it. You can do it. There's no difference between a male or a female. We're all scientists there, and you can see a lot now with the COVID pandemic. There's a lot of scientific community getting together. These are female, males, it doesn't matter. The gender really doesn't matter with science. So I would say, yeah, the sky is wide, the sea is wide, follow your dream and do it.
So when did you realize that you want to do this area of research?
That goes back into actually my undergrad, third year pharmacy. I did a pharmacy degree at UCL School of Pharmacy. So I was taught at the time by Professor Kostarillas, who ended up being my PhD advisor. So yeah, it was fascinating because we study in pharmacy, you study about chemistry, pharmacology, biology, and we study as a pharmacist, we study a lot about formulation, how we make drugs into tablets, how we make them into solution, and so on. So one of those exciting area at the time was drug delivery. So I remember Professor Kostarillas talking to us about the movie Fantastic Voyage, as I would say it in French, and that's where it got me a bit excited. And then I approached him to do my MPharm master project at the time, which then I did my mine with him, and I continued my PhD under his supervision at the time at the School of Pharmacy.
Thank you very much. Well, it's really lovely to have you here today, and we wish you all the best in your future work. Thank you very much again.
Thank you.
Now we will move to our next speaker, Helen Howard. She's a PhD student at the University of Nottingham. and her talk today is about how does our brain know we have a body, and how does this affect how we socialize.
Hello, my name's Ellen and I'm a PhD researcher from the University of Nottingham, and I am fascinated by the brain. But more specifically, I'm interested in the relationship between our senses, our bodies, and our social interactions. So I'll just start with a simple question. How do you know that you have a body? So you might have said, well, I can see it and I can feel it. And that's exactly it. Our brain puts together information from lots of senses in order for us to feel like we have a body and to know where it is in space. So as I said, it might use visual or touch information, but there's also some other senses that the brain might use. So one is interoception, and that is awareness over internal bodily processes. So that might be knowing when you're hungry or being able to feel your heartbeat without trying to find your pulse. And another is proprioception, which is the awareness of where your body is in relation to yourself and to space. So there's an easy way to demonstrate this. If you put your hands behind your back and close your eyes, and then you have to get your finger to your nose. So you should be able to do it. And that's because the brain is using proprioception to know where your finger is in relation to yourself and the rest of your body so that it can easily and quickly find your nose. So the brain uses information from lots of different senses. But the key thing to really feeling like this is your body and you feel like it's yours is when the brain combines these senses together. And when it combines these senses, it's called multisensory integration. And really it is this integration of all this information that really makes it feel like you have a body, you feel like it's yours, and you know where it is. And then it's these feelings that allow you to navigate the physical world and be able to move around in it. What is really fun is we can take advantage of how the brain integrates sensory information to create body illusions that help us understand a little bit more about how the brain works. So at the University of Nottingham and Loughborough University, we use augmented reality to create body illusions. Let's take a quick look at an illusion that makes it feel as though your hand has disappeared. So what we can see here is an augmented reality system called Mirage, created by Dr. Roger Newport at Loughborough University. And what the person has to do is keep their hands in between the blue bars as they start to move. Now, as they move, the image of their hands is actually slowly moving inwards. And this means in order for their hand then disappears from view and they use their left hand to try and find it, it's not where they thought their hand was. Let's have a look at a real person's reaction to this illusion. As you can see, she is really surprised when she reaches across with her left hand to not be able to feel her right. And that's because her right hand is so much further over than where she last saw it to be. So it's that combined loss of visual and touch information that really makes it feel as though your hand is missing. So that illusion, although it's very fun, does also show us how the brain integrates visual and proprioceptive information in order to make sense of the world and know where our body is. But even if you don't have an augmented reality system lying around, you can still have fun with some body illusions at home. So let me show you just a few. This first illusion is called the Anne Boleyn illusion because it can make you feel as though you've got 6 fingers. So all you need is a mirror and a willing volunteer. What you want to do is set up a mirror in between the hands of your volunteer so that they can't see their left hand and they can only see their right and a reflection of their right hand in the mirror. Whilst your volunteer is looking at the reflection of their right hand in the mirror, you're going to stroke each finger on both of their hands in time like this. You can do this a couple of times, and then on the final time, we'll make it feel like they have a sixth finger. Now, when you get to the little finger again, on the hand they can't see, you need to stroke the inside of the little finger, and on the one they can, you stroke the top. Then you stroke the outside of the little finger they can't see and draw an imaginary finger on the hand that they can. This illusion works because your brain is combining that touch information on your unseen hand in combination with the visual of seeing an imaginary finger being drawn. So that's what can make it feel like you have an extra finger. For this next illusion, you can have a go yourself, or you can rope in your happy volunteer again. So all you need to do is stretch your arms out, turn your palms outwards, cross your hands over, and put your hands like this. And then you want to try and pick a finger to lift up. So I'll try and lift my right index finger. And then it's much harder to try and pick the right finger. And that's because of the unusual position you're putting your hands in, sort of messes up your senses a bit and makes it so much harder for your brain to try and work out which finger it needs to lift. So have a go yourself, or get your volunteer to have a go and see how well they do. This last illusion is really quick and simple. So all you need to do is cross your index and middle finger over like that. And then if you run it down the center of your nose whilst your eyes are closed. So you just stroke your nose like that. And if you keep doing it, it can sometimes feel as though you've got two noses. And that's because your brain is getting information from the inside of both of your fingers along the bridge of your nose. So it can sort of trick it into thinking that there are two noses there. So have a go and see if you feel it too. So what can these illusions tell us? Well, in a research context, body illusions can help us understand how the brain integrates senses, and can also help us answer questions like, how do children develop a sense of bodily self? And how might it impact upon their social interactions? So I've already spoken a bit about how sensory integration is so important for knowing where your body is and therefore being able to move around physical space. But we also think that it might be really key for how we socialize. Think about when you're listening to somebody talk. It's normally a lot easier if you can see their lips move as well as listen to what they're saying. And that's because the brain is integrating that visual and auditory information to make it easier to understand. So sensory integration is also key for our social interactions. In some recent research that we conducted at the University of Nottingham, we wanted to investigate the link between our bodies and how we socialize. So we asked people to complete a task with a stranger where they either had to tap in time with that other person or tap in time with the image of a bouncing ball. And what we found is that people were significantly more likely to want to affiliate themselves to that stranger after they'd been moving in time with them. And what we think this shows is that when we coordinate our movements with other people, it creates this sense of similarity, which makes us more likely to like them. So integrating your senses, creating this sense of bodily self, allows you to move in time with somebody else, which could make you more likable. Sensory integration is therefore fundamental, not only for navigating the physical world, but also the social world. So next time you're having a chin wag with someone, think about just how important your senses and your body are in social interactions. Thank you so much for listening. And if you have any questions or you'd like to know more, you can email me at my email address below.
A very interesting talk from Alan. Thank you very much for having you with us today.
Hi.
Hi. Nice to
thank you so much for having
you
me.
with us today. I'm just looking at the comments to see what people are asking about your talk. And I also would like to remind our audience to keep posting your comments either on the YouTube or on the Facebook stream, and we will have the chance to answer them live with the speakers, or if not now, then they can respond to your questions later on. So one question people are asking, how are the somatosensory effects useful to us?
So this sort of research and these illusions do have some really cool applications. So one of them is to understand how we process pain. And we think of pain being very physically based, but actually it is sort of a creation of the brain. And a really good story that demonstrates this is a few years ago, we had somebody come into the lab to do some testing in the Mirage augmented reality system, which you saw in the video. And it was an older lady, and we were going to do an illusion on her where we gently tug on her finger and the visual shows her finger being stretched. And it really makes it feel as though your finger's been stretched and you've got this massive long finger waving around. And she said, okay, so you have to be a little bit gentle with me because I've got quite bad arthritis. And she put her finger in the Mirage, and we did the finger stretch illusion. And she said, oh, that's really weird. My pain's gone. And so that's just one way that we think that it could have a really interesting and cool application, is for these sorts of treatments of chronic pain. And then there's also some of the research that I'm doing is working with autistic individuals as well, and trying to use these illusions to understand how sensory information might be put together in a way that makes it uncomfortable or not a great experience for the individual, and trying to put support there for somebody if they have, you know, quite uncomfortable sensory feelings. So there are like a couple of the ways that we can use those illusions and stuff.
Yeah, that's quite interesting, and lots of interesting tricks. I'm sure people will be happy to try them afterward. So do you think this could also be useful when there is a damage to a certain brain area, and maybe help to improve brain plasticity?
Yeah, that's really interesting. And I think this would relate a lot to what can be found when people have amputated limbs. And you might be aware of the phenomena of a phantom limb syndrome. And that's where somebody, although they've had maybe their arm amputated, they really feel like the limb is still there, and they can still feel pain, which is obviously not great for day-to-day experience. You don't want to be experiencing pain. And things like this can be used to almost give that feedback back to the brain to alleviate that pain. And so you can use like a mirror box. So like with that Anne Boleyn illusion, you can have put your hands either side of a mirror, and where the amputated limb is, you can have the reflection of your other hand. And you can do exercises like opening and closing your hand, and that can release some of that phantom limb pain. So I think it definitely could be used for sort of brain area damage or physical damage to the body, and help the brain sort of get used to changes either in the brain or the body.
Okay, interesting. One of the questions people are asking, why do some of these illusions work on some people but not others?
That's a really interesting question. And I think this is definitely a difficult one to answer conclusively. I'm definitely somebody who really experiences the illusions. So like the person that you saw in the video, I will sort of squeal and scream out when I feel like my hand has disappeared. I think part of it is to do with how the brain is putting together that information. And if you are using that visual information and that's being quite dominant in your processing, you're probably more likely to fall for some of those illusions. Whereas if you're really, really aware and your proprioception is really, really high, so like in the disappearing hand, you're not necessarily going to feel that the same, because you've got that awareness that your hand is not where you saw it to be. So there's probably some like nuances in what information people use and in what context as well.
Okay. We have another question. People are asking, does this similarity of movements help people feel closer together when they are dancing together?
Yeah, so this is a really cool phenomena of the role of our bodies and how we move them and how we feel close to people. And I definitely think it would be related to when we dance with somebody that you might feel close to them, because that's what our research has basically shown. When you move together with somebody, you're more likely to like them, to want to be affiliated with them. Other researchers found you're more likely to help that person, cooperate with them if you're moving in time. So I definitely think that dancing with somebody could boost how much you like them or how much they like you.
Yeah, probably it would. Another question, do you think your work could help applications for patients with strokes during rehabilitation?
Yeah, definitely. And it's these sorts of illusions that have been used, and this sort of research that has been used in a pain context. So the lab that I work in, we don't focus on pain as much. But I did at the beginning of the year do a research visit in Australia at the University of South Australia. And the lab that I worked with there, they focus on pain. And so they're trying to work with physiotherapists to sort of change our perception of pain being this physical thing, and there must be something physically wrong with you in order to experience pain, which just isn't the reality of it.
Yeah.
So I definitely think that this sort of work has huge impact in people's experience, and to try and stop people experiencing chronic pain like that.
And another question, can you use it together with cognitive behavior therapy for chronic conditions like tetanus?
Interesting. I don't know. I mean, I would guess that it would work together, because that sort of multi-therapy approach is usually the most effective. So I can't see why not, but I don't I don't actually know if there's been research into that or not.
I have also some questions for you. So what makes you decide to specialize in this area?
Yeah, so well, when I was younger, I didn't really have any expectation to go to university. It wasn't on my radar. Both my parents are entrepreneurs, and it was definitely more of, oh, when I turn 18, I'll try and start my own business. And it wasn't really until I was 14 or 15 and I took part in this extracurricular sort of workshop at school. And one of my teachers did a whole workshop on psychology, and I was like, oh my God, this exists. Wow, okay, people focus their lives on researching this. This is awesome. And yeah, my mum will say that I came home in a very dramatic fashion and said, okay, my whole life has changed. So from that, then I was really motivated to pursue psychology in general at university. And then whilst I was doing my undergraduate degree, I worked with the Mirage Lab, doing an internship and some public engagement work with them. And I just thought it was so fascinating how much our senses and our body relates to so many different things. And it felt like such a new and interesting area that I couldn't not pursue it.
It's very exciting area, and there are a lot of unanswered questions there.
Yeah, definitely.
I'm sure that there will be a lot of research ongoing on that in the future. So do you have any favorite experiment?
Oh, that's a tough one. I do really like the one that I spoke about briefly in my talk about the link between synchronizing with somebody and the social benefits, because that was a really interesting study to take both sides and see about the sensory and sort of perceptual processing side of being able to move your body in time better to somebody else than you can with something that's non-social, like a bouncing ball. But also then to see the consequences of it, and that directly after you can see that children are more bonded to the stranger and adults as well. And also that children in an interaction following that tapping task, they were more likely to mimic that individual as well. which you think then sort of feeds back, because then they're moving more similarly, and it's sort of this loop of your senses, your body, and your social, like all looping together. So that was really interesting to get sort of both sides. So yeah, I would say that that has been a really interesting one I've done recently.
Mm-hmm. Well, yeah, it sounds like a quite a tricky one also to do.
Yeah.
Well, thank you very much for being with us today, and wish you all the best in your future.
Thank you so
Thanks
much.
a lot.
Thank you.
So now we are moving to our next speaker, Dr. Sana Pamel. And Sana is working in digital research at the University of Nottingham. And her talk is about creating alternative realities for research, virtual, augmented, and stimulated.
Hi, I'm Sana Pamel, and I'm a digital research specialist at the University of Nottingham. So a part of my work is to support researchers with digital technology. And today I'm particularly talking about the type of research that involves people using some form of innovative technology or service, some of which may not even exist yet. So such research looks at questions such as how do users interact with the technology? Do they accept it? Are they able to safely operate a new device? And some research tests training for situations that are dangerous, or training for using a technology that doesn't yet exist as well. So basically, the problem is that these questions are impossible or very difficult to answer in real world settings. But there are actually also situations where the product exists. For example, if it's like a new type of beer or pasta sauce, and testing those in a real world setting can be difficult because there's hardly any control of the different conditions. So one participant might be in a completely different condition than another participant, and that may affect the results. And actually, currently we have the situation with COVID, where a lot of research is quite difficult to do. Generally, research involving participants, contact with participants, is difficult to do with social distancing. So laboratories can solve some of these problems. So, for example, I mean, we can test the flavor of beer or pasta sauce in the same settings for each participant each time. However, a lab can be quite different from a pub visit with friends, for example, or a holiday in Italy. You can control all the variables around the environment, but it can just be too unrealistic and too sterile. So as a solution, I'm talking about simulation and expanding this into augmented and virtual reality as well, which can give researchers the opportunity to have cost-effective experiments, have them safe and controlled, and have innovative experiments as well, or test innovative technology that doesn't exist yet. So simulation, according to the Oxford English Dictionary, is to give a false impression of something, to reproduce the condition of something, or to look and act like something else. So according to Wikipedia, it's the imitation of some kind of real thing, state of affairs, or process. So a simple low-level simulation can be just a really simple mock-up made of a set of papers or cardboard. And with that, you can already get quite some good insights into whether a user is able to find out which button to press also to solve a particular task. And that's really at a low cost, to get some really valuable information at a low cost very early on in the design process. And then for more elaborate research, we can use simulators of cars, of trains, surgeries, ambulances, and a lot more. So for example, a typical driving simulator consists of a body of a real car, and then you surround that car with screens that show the road environment, and that's already giving quite a high fidelity, like medium to high fidelity research environment, and you can answer loads of research questions with that environment. For example, with cars, looking at novel navigation systems, looking at automated cars, and basically anything that hasn't yet been produced at a high, at expensive investments. Augmented reality is defined as you experience the real world environment, but enhance objects in the real world environment with computer generated information. And that information can involve different senses as well. So augmented reality itself can be simulated. So for example, like within a simulated environment, like in a driving simulator, you augment objects in that environment. So for example, a lane on the road, you can highlight the lane and use that to help the drivers navigating to wherever they want to go. And AR, so augmented reality, can also be used for, especially now, for remote laboratories and experiments, but also to visualize the results. Once you collect all the data with experiments, you can use augmented reality to visualize the results. And virtual reality is the computer-generated simulation of a three-dimensional image or environment. So you basically simulate an entire three-dimensional world, and the user can interact with the world, I'd say almost in a real way, or some kind of physical way, for example, with some kind of controllers that they hold in their hand. And the participant, in order to see that world, wears some kind of goggles, helmet, that has a screen inside. So, for example, colleagues of mine at the University of Nottingham, they simulated a stressful restaurant kitchen environment called Corrupt Kitchen. And in there, conflicts are created deliberately with time pressure, and then there is a staged opportunity to bribe an inspector, because, if it's not possible to keep the kitchen clean, there's an inspector, and then you have the opportunity to bribe that person. And others have simulated emergency situations and then observed the responses of the participants. So then both are done in order to create training for people to better prepare them for those situations. And the virtual reality in those cases allows testing the situations with people without actually really placing them in such situations. So as a conclusion, in this way, simulation, especially augmented and virtual reality, can allow cost-effective, cheaper, safe, controlled experiments, and really innovative experiments of things that don't exist yet. So, thank you.
Thank you very much, Sana, for a very interesting research area. I would like to remind everyone to keep posting your questions to the speakers on both the YouTube and the Facebook stream. Hi, Sana, how are you today?
How are you? How are you, Sarah?
Thank you. It's nice to have you with us today.
Thank you.
I'm all right, thank you. So one of the questions from the audience asking on how much do you have the chance to completely reinvent the way we interact with things? For example, in a surgery.
That's an interesting question. I think the point of simulation is sometimes to do things that we that we actually need to reinvent, so to say, that to do things that are not, or to kind of interact with things that are not there yet, not developed yet. So you can simulate something and then work with that. Oh, is the question also related to my everyday work? I think that's a bit more complicated. I'm actually not a researcher anymore. I support researchers, and yeah, there's lots of really interesting stuff going on. So
I guess the question probably is asking more about how could you use augmented reality to help training people, maybe in the surgical field or something like that. Do you think that would be useful to change the way that we do things and maybe move them to another level?
Mhm. So I personally haven't worked in surgery, but I know a really good simulation lab, also at Nottingham, and that was really interesting. So they have like a lab set up with more or less, I would say, dolls, like so they look like humans, and you can kind of operate on them. But it's not real. And then there's a control room where things are manipulated, and they say they simulate a medical condition or something, or some kind of crisis, some kind of problem or so, and then the students have to solve that. And I guess, yeah, all of the this is like the simulation where you have like I'd say a robot or some kind of thing, or a doll that you can touch, and you can work with real instruments on these kind of fake humans. But you can also yeah, probably also just as well use virtual reality or augmented reality in ways. I mean, for example, augmented reality can probably help you see things through like the surface of a body, for example. That's something I can imagine. But I personally haven't really worked with AR or VR in that regard.
You mentioned the example of the busy kitchen and how augmented reality can help to simulate this. And I would imagine in the future there will be more and more examples that are will be happening. In terms of research, how can you give us like more example where you can use augmented reality for collecting like data, or what kind of research do you think this would be more applicable compared to others?
So do you mean particularly augmented reality? Because the kitchen, for example, that was a virtual reality environment.
Yeah.
So that was for training. That was done for training, to learn with training, and I've worked with similar I've seen, so to say, similar research with emergency situations and training for those. So that was really interesting. The actual research I did with augmented reality was more in terms of driving. And we were actually, I'd say, simulating virtual reality. So we had a simulated driving environment, and we were in different ways kind of augmenting things in the road scene. So, for example, the lanes you're driving on, or even projecting a car onto the windscreen. And that car was driving in front of you, and you had to kind of follow that car in terms of navigation.
Interesting. We have another question from the audience asking, how did you find the transition between working in a research towards supporting research?
I think it was quite interesting. This sounds more extreme than I would say than it is, because before my PhD I was working in IT as well. I was a software developer for several years. So I kind of felt that I reconnected with that past, so to say, so with that side. But also I really appreciated working with more research. You know, I couldn't like do the research myself anymore and really dive into like one topic, but I could connect with lots of different researchers and different very different topics. And I definitely found it really interesting to have so much different stuff going on at the university.
Interesting. So what was your favorite subject when you were at school, and how did you get into this area?
At school, I think one of my favorite subjects was actually maths. And I had a really good math class towards the end of my school time, with a really like a teacher who was really passionate. And I mean, I also liked, physics and so on. So I had this kind of affinity towards technology, I'd say, in my studies. But still, it was very hard to decide because I also like languages. I really liked studying French, and I also found politics really interesting. So I actually studied business and economic science first with a major in information systems. So that's why I'm kind of in between things, and I feel like I've been oscillating in between technology and people, if that makes sense, just because I couldn't really decide on that one thing.
Interesting. So I think that these two subjects probably helped you in your area. One last question: how many women are there in your field, or is it mainly dominated by men?
What means in my field? So, in engineering, there've been it's quite relatively dominated by men if you look at the total numbers. But in human factors, where I actually was when I was still a researcher, there were more women, and with the students you could see more and more women coming, and that was very good, very very encouraging, and also for me, I find it very encouraging and how can I say, motivating when there are women in management positions. So to say, above me in the hierarchy, when they're women, and they've been there all along. So when I did my PhD, and then when I was working as a research fellow in Nottingham, and now as I'm in information services now in the university, they're always I'd say really good female role models, and that has been, I would say, surprisingly important for me in during my career. I didn't expect that when I was still a student, that I could actually play such an important role.
Thank you very much for having you with us today. And now I would like to introduce our next speaker, Dr. Lucilia Rodrigues. She's an associate professor in sustainable design at the University of Nottingham. And her talk today is about climate change and your home.
Hello, I'm Lucilia Rodrigues. So much has changed since I signed up for doing this, and I really wish you were here with me at the Market Square in Nottingham, where I'm not standing in a SoapBox box because I haven't got one. But I thought it was suitable for me to talk to you from here. And it's such a shame I can't see you in person and answer your questions. Maybe send them my way, and I'll answer them by email. So I started by showing you Nottingham's commitment to become carbon neutral by 2028. And so much has been done in relation to climate change in the last few years, a new climate emergency that became really headlines. But it's quite difficult to understand what's the relationship between that and your home. What can you do to make your home better? Well, that's what I'm here to talk about. How does your home relate to climate change? It's easy to think of reaction to climate change in a home by thinking about energy efficient technologies alone. The first thing that you should be thinking about, and is the most important thing, you already know, is how to reflect your climate conditions and how to make your home more comfortable. By making your home more comfortable, you need less heating or less cooling, and therefore you're going to impact less on the climate. And how do you know what to do? Well, think about what you would have done if it was you. So, for example, I was just standing outside in the Nottingham Market Square and got a bit chilly. So I've actually come and took shelter in the Nottingham main council house. Now I'm protected. I'm protected from the wind. Hopefully you can hear me better too. But what I have what you would have done to your home. So if I were to design a energy efficient home in a climate like this, I would have been thinking, it's a bit cool out there for a great part of the year, and therefore I should be trying to protect my home from wind or cold wind in particular. In this case, in Nottingham, from the north wind. The other thing I'll probably think about when I'm out here is to wear a jacket. Well, same goes for your home. If you live in a cold climate, or if you spend some of the year in a cold climate, your home needs a jacket. In the case of buildings, that's called insulation. Insulation is nothing else than fibers of some sort, some material of some sort that can keep bubbles of air inside, or the majority of insulations are made of that, and therefore they has less heat transfer. It's very much like a woolly jumper. Well, it's a bit sunny again here, so I'm going to step out into the sun. And that's what you do with your home. If it's sunny and if you can make any use of that, step your home out into the sun. Of course, you probably can't move your home right now into a sunnier location, which would be lovely. But you can perhaps open up the windows, open up doors towards the direction of the sun, and enjoy the maximum sunshine you can by avoiding having any surroundings that might block the sun coming to your home. Now, if it gets a bit too much, is it too hot? And a lot of us experience overheating in the UK these days. Well, close it up. I know that's a counterintuitive thinking. I say it gets too hot over the day and you're getting too hot in your home to sleep at night. Well, when it's hot outside and inside, close your windows. Close your home to the excess sun and excess heat and keep it cool. Then you ask me, what if I can't? What if I can't insulate my home? What if I can't open more windows or less windows and I'm limited by my space and my location? Well, in that case, you should be thinking about energy efficient technologies. You should be thinking about improving your heating system, improving your cooling system if that's the case, and try to use as much as possible renewable energy. Now, I hope that gives you some ideas. It's a shame I can't hear your questions right now, but I'm really looking forward to listening to some of them, maybe via email or via chat soon. Thank you.
Thank you very much, Lucilia, for the interesting talk. And thank you very much for being with us today. Very interesting area and quite topical research. So I have a question myself about what kind of advice do you give for people who are living in old-fashioned houses, and how can they improve to make it more sustainable or energy efficient?
That's a very good question, because older houses are much harder to improve than newer houses, or if you're building a new home. If you have an older house, particularly if it's in a conservation zone or something with a historical importance, or maybe even listed, then you have a lot more limitations to comply with, and that makes it a lot harder. Of course, the first and foremost thing to focus on would be to improve the fabric. So if you can change your windows, if you can afford that, add insulation if that's permitted. Ideally, external insulation or cavity insulation. If that's not possible, then you have to look at internal insulation, which are more expensive and harder to resolve. So those are not very easy properties to resolve. We also, of course, can look at installing renewable energy. So if you have enough roof space that face somewhere close to the south, it might be worthwhile and cost-effective to add renewable energy such as PVs or solar hot water. But I would say that the first investment should be on your fabric, and the second on your own habits. Look at what you do at home and see what it can change. Something the things you do can avoid windows being open on the heating zone, for example. Try to avoid the front door being constantly open and closed without having a lobby, and other things like that. So try to keep your temperature maybe a little bit lower at 20 degrees than 22, and that would make a big difference at the end of the day.
Mm-hmm. Interesting. We have a question from the audience asking, how can we learn or how have you learned from other countries or other cultures, other places in the world, maybe things that are more counterintuitive that have been working or in practice for other places, such as hotter places, and how can we adapt what they have done in our culture?
So, for example, it's very common to see in hotter countries, both in Europe and other parts of the world, so to the Middle East and parts of Latin America, shutters on the windows, and the windows being shut throughout the day, especially throughout lunchtime and the early afternoon. And I've heard before, even my own sister once said, we were in Italy and it was a very hot summer, and she said, "Oh, look at those houses, it's very hot and all the shutters are closed. What are they doing?" Well, they're doing the right thing. Because when it's too warm outside, even though you might be thinking it should be ventilating, if you ventilate, you're just welcoming warm air into your home. And you also might be welcoming sunshine at the time when you don't want it. So things such as shutters, masharabias, which are ways of allowing ventilation, but still screening the sun, leaving the sun out, are things that we can definitely import from other countries. And I think my suggestion is always to look at vernacular architecture and vernacular strategies, because before there was air conditioning, before there were heating systems, people still survived in their homes and lived quite comfortably. So they just looked at strategies that were, as we call, passive strategies that worked with their climate. And we tend to have forgotten a lot of those because it became so much easier to have mechanical ventilation, mechanical conditioning systems.
That's, yeah, very true. We have another question. Is there anything that we have learned from how animals deal with the extremes?
That's an interesting question too. I actually use some examples in my lectures. For example, I use the example of elephants. Do you know elephants have a lot of insulation, right? They're quite big and they have a lot of fat. However, they also have very big ears, which they're often moving about in the wind. And the reason is because that whole surface can exchange a lot of heat to the environment. There's no fat on the ears, and the blood goes around the ears very close to the surface. So by doing that, it's cooling its own blood and keeping its body cool. Isn't that an interesting strategy? So it's too hot, let me allow myself to cool down by experience of enhancing the wind. Another one that I use very often is a chameleon in the Namibian desert, which is a fantastic example of survival in a very extreme climate. So we're talking here extremely high temperatures in the day and extremely cold temperatures in the night. So a chameleon has to survive in those temperatures. And what it does, first thing in the morning, it actually stands in the sun and makes its skin very, very, very dark, so it can absorb as much sunshine as possible. When it starts to get hot, it turns its skin white, and it also moves quite fast, so it doesn't get burned on the grass, on the sand. So it is a fantastic way of adapting to such a climate, when such a harsh environment, with a very simple strategy. You're just changing the color, absorbing more heat or releasing more heat when you don't need it. I think we can import all those learnings to building design quite easily. And as I say in my video, and I also tell my students in my lectures, is that you actually intuitively, we already do a lot of things that I'm teaching them to do in buildings. It's just that we tend to see buildings as such more complex elements, but in reality, if it's a cold environment, close up. If it's a warm environment, maybe open up, welcome the wind, or get away from the sun, the things that you would have done if you're personally in that situation.
Mm-hmm. So if you have the money that is available in the world, what would be your favorite sustainable home design?
Well, I hadn't thought of that before. In fact, if I didn't need to be working, I'd probably be in a warmer country because it's a bit easier not to have to go through a very long winter every year. Not that it's an extreme winter, but it's long enough and dark enough. I think what I would always like to see is a home or generally a building that works with the seasons and changes and is alive and is responding to what's happening in the environment and responding to the functions. In a home, I would like to see a home where in the winter can be cozy in smaller rooms or less rooms they would normally use in the summer, that you can welcome some sunshine through the window so you can sit in the sunshine and keep the home warm. As the spring comes, maybe you could expand into another zone of the house where the temperature is less controlled. And as the summer comes, you can expand into a larger outdoor garden and your home can become the whole space, including the outdoors. So I think that idea of expansion and contraction for me is very attractive. I think an idea that a building is responding constantly to the external environment and to your needs is really attractive in the home.
Mm-hmm. Interesting. So why have you decided to become an academic and more to specialize in this area?
So I'm an architect by background, and I worked in practice and designing. I designed a lot of homes back then, and I worked in a few big commercial buildings too. And I always thought I needed to learn more about sustainability, and I couldn't quite find a way of doing that. And I'm from Brazil, without having to stop working or being able to afford with the work. So eventually, I got a scholarship to come to the UK, where I did my master's, and I felt that the more I learned, the more there was to learn. And I think if you are at the moment studying towards a master's or towards a PhD, you're probably experiencing that. The more you learn, the more you think, I don't know enough. I need to learn more. So through that, engaged in a PhD at the University of Nottingham. And when I came towards the end of my scholarship funding, I applied for the job, for the academic position, thinking it was a temporary decision. And as soon as I started the job, I realized that teaching had a lot to do with me. And it was quite a moment when I realized I had been in academia, so being a full-time academic. I'm a professor in sustainable business at the University of Nottingham since 2008 now. So it's well surpassed my time in practice. I know I'm much more of an academic now that I'm an architect for my practice, although I do have the practice experience. And I think that really suits me because I love teaching sustainability and I love making a difference. And I know a lot of my students are out there now and they make a really big difference. I'm always hearing back from them with the wonderful things they've done and how they're changing their own countries. The majority of them being international students, so they are spread across the globe and they are really making a difference. And that feels great.
Fantastic. So in the courses that you teach, are there a lot of girls, or how do you see it?
It's a great question. It's great to see the SoapBox Science focus on researchers, the female researchers. My department is a very old department. It's 177 years old. It's called the Department of Architecture and Built Environment. There's never been a female professor before me, so I'm the very first female professor, which I'm both proud and embarrassed to say. Yet 60% of our students are female students. Where I came from, generally about 70% of the students at the university level are female students too. So I think things have been changing. I hope this is just the starting of a whole new cohort of professors, of female professors in our department, because I think it's very important to have a role model that you can look up to, and you know that despite your gender, you still have the same chances as everybody else, and you have a good position to look up to. But it's a very interesting question. I also heard that, in fact, in practice, of people who come out of the university, have done architecture and go into practice, much more likely to be male than female, even though the students are more likely to be female. And also the top of the classes in general are the female students. So it is a very strange world, but I hope that's changing and we're going to have a better balance in the years to come.
That's really interesting to see that things are changing and more females are taking or getting into that area of research. So what advice do you give to academic females or to female scientists to make them continue that area of work?
So I think in research in itself, there is a lot more male than female still, and in terms of leadership and is generally much more male than female. So I think that's sad, but it doesn't mean that there isn't space for the female scientists to come up to. And I hope you can see the fact that I'm a professor in this area is just one of the many that could be there, and you have all the chances to get to that stage. Sadly, I do feel that many females work harder to get to the same place, maybe because we feel the need to prove ourselves, maybe because we always believe we can be better and we don't actually push ourselves forward as much as it's just a part of the culture, isn't it? It's not really intrinsic. It's not really a personality thing. It's more to do with the fact that we don't feel we're ready for the next step, they're ready for the promotion. But just prepare yourself always with that in mind. So when you look at promotion, when we're looking at even applying for a career, already think about what is the criteria, what do I need to move up to the next stage. If you are in academia and if you want to become a professor, the best advice I've been given, and I'm going to repeat here, is what you want to be a professor of. Know from the beginning and work towards that goal. So you know that all your work and all your effort and your energy are going in the direction of making that possible. As a professor, you're meant to be the person who is recognized as the top scientist in that area, and is a big place to take. But the more you work with a direction, something that you really have a passion for, potentially and hopefully even aligning your research with your teaching and making sure things progress hand in hand, the more chances you have to be the big person known in that field. And that's what it takes to get that stage. The one people are going to come to ask the questions.
That's a really very good advice. And it's really good to be focused and have a clear dream to work toward. Any other advice that you would like to give as a female scientist? You describe yourself also as an artist before you moved to being a scientist. So how do you see yourself?
Yes, so I think, as a female scientist, you always depend, especially I'm still based in engineering, as I know my colleague who just spoke before me was, and we are mostly surrounded by male scientists and male role models, which is both a good and a bad thing, because a lot of them are very inspirational and I do look up to them. But I also feel that it would be nicer to have female scientists that I can always look up to, and some female leaders that can accommodate. I think the thing that has been very interesting for me to watch in this last year, where things have been very hard for everybody, hard for countries, hard for every household, and hard for the institutions, is that a lot of the female leaders have been the highlight as delivering what we need really at this moment of pressure and difficulties. And I think that's an eye-opener for everyone as a society. So it's great to watch that happening, and it's great that they're proving themselves as the leaders we want. And a lot comes from being able to sympathize and being able to be kind and be able to be emotional, as well as being assertive and decisive and actually pushing things forward as they should be. So I don't think there is if you ever cried and you ever felt embarrassed for that, don't, because that's what makes us better, is the ability of embracing that emotional response and dealing with it, and then asserting or rethinking a response to the problems based on both that emotional connection as well as the practical needs.
Yeah, that's very true. Well, thank you very much, Lucilia, for being with us today and for sharing your science with us. So we are coming toward the end of your SoapBox Science Nottingham event today, and I would like to thank all the speakers for their lovely talks and for the interesting discussions. And well, I'd like also to thank my colleagues, Rebecca and Helena, for organizing this interesting event. Thank you very much for your time. And just one more thing, would like to remind you, if you have any other questions, please either post them on SoapBox Science Facebook page, or on the YouTube, and then the speakers will come back to you with the answers. Thanks again for your time, and wish you a lovely afternoon.